I was recently sharing with my friend about some of the work and interest I have in computational biology, and she asked me, what exactly is computational biology? I think one of the ways that I can explain it well is by illustrating with an example of why this field is so useful. And I think in this video I want to highlight a specific example from my own real experience to show why computational biology is a worthwhile thing. When we come to thinking about this realm of epigenetics, where we look at not just the sequence of DNA, but also other things that are inherited to other generations, one of the things we look at is something called DNA methylation, specifically 5-methylcytosines. And what on earth is that, Daniel? You may be asking. Well, let me explain. Basically, when it comes to DNA, we have 4 nucleotide bases, A, C, T, G. And one of those, C, or cytosine, can have a methyl group. A methyl group is going to be carbon attached to 3 hydrogens as long as the carbon is attached to something else. Otherwise, if carbon is attached to 4 hydrogens, that's a methane. So all that to say, sometimes a CH3 or methyl group is attached to cytosine. And that's something that we call DNA methylation. And it turns out that those methyl groups can play a big role in epigenetics. One of the things that's really interesting to us as computational biologists and biologists in general is assessing whole genome methylation status of different organisms, especially under different environmental or genetic conditions. And one way that we really do that is by using something called whole genome bisulfite sequencing. This is a really cool technique for assessing DNA methylation globally, that is across the whole genome. And the way it works is doing something called bisulfite converting DNA. Really all that means is that we're going to use a technique to convert methylated cytosines into uracil, which after PCR or a polymerase chain reaction, we're going to get thymines. And I know this is a lot if someone's not trained in biology, but basically what I'm trying to say is that biologists are interested in this question of methylation, and we have this biochemical technique to assess it. But just because we can biochemically convert methylated or unmethylated DNA into something useful for us, that still leaves the problem of analyzing that huge metadata. And to do that, we need really good algorithms to parse out all these different sequences that we get from reading a large population of cells for their DNA after bisulfite conversion. And those algorithms require good computational minds to come up with. One really good package that's widely used for whole genome bisulfite sequencing is called Bismark. This is the one that I used for doing 2 things, whole genome bisulfite sequencing and something called chip bisulfite sequencing. Unlike whole genome bisulfite sequencing, in chip bisulfite sequencing, basically we're going to look at specific regions of the genome that are bound by a protein of interest, cleave away those fragments, and then sequence just those fragments. That allows us to get a more targeted understanding of the methylation at those protein binding sites. So I did a Bismarck analysis of both kinds of sequencing data, and what was really cool is that over the course of several weeks, I used the NIH supercomputer for a total of over 40,000 CPU hours. There were literally like tons and tons of computational resources used to analyze all this data, and then it ended up allowing us to have some really interesting results. So why do I say this as an example? A, it was really fun for me to go through this process, and it really helped to further confirm the need for computational biology, because basically you need to know the biological techniques and the biochemistry, the mechanisms going on, but you also really need good computational resources, in this case, high performance computing resources, and good computational algorithms to be able to parse out all this immense data into something meaningful. And it turns out that computational biology, in all its broad areas, ends up having very similar benefits where, yeah, you need to know the biology, but you also need to have good algorithms, good computational packages, and really good computing resources to be able to do your work. And really, this kind of analysis is only possible because we have really good cutting-edge molecular biology and we have really good cutting-edge computer science. So hopefully this real example from my life can help to illustrate a little bit of why computational biology is such a worthwhile and interesting area.